Okay, so welcome to this next video in which we are discussing leukocyte extravasation. Okay, so we're just about to discuss how type 1 activated endothelial cells recruit neutrophils. So we've seen that type 1 activated endothelial cells start releasing prostacyclin and nitric oxide, which causes vasodilatation, and they also contract to allow the buildup of an inflammatory exudate. Now let's see their involvement in recruiting neutrophils. Okay, so, new piece of paper. Right, so type 1 activated endothelial cells start to put two molecules on their surface, okay? Now, one of them is a protein. Now, you might think, well, how do they put a whole new protein on their surface so quickly? Because remember I told you type 1 activation takes minutes to occur, okay? So if they're not, if it's taking minutes, then they can't be synthesizing this protein from scratch. And indeed they're not. They already have the protein. It's stored in vesicles within the cytoplasm, okay? Now, normal endothelial cells do not have the protein on their apical membrane. Instead, it's all stored within these vesicles here. So you have many vesicles, each with many copies of this protein in. Now, what is this protein? This protein is a protein known as P-selectin. So it's a selectin cell adhesion molecule. Okay? So, this little red protein that I'm drawing in here, this is known as P-selectin. Okay? Now, these vesicles which sequester and store the P-selectin, these are known as viable palade bodies, and all endothelial cells have viable palade bodies. So that when they undergo type 1 activation, what can happen is they can fuse these viable palade bodies with their apical membrane and release P-selectin onto their surface. Okay? So here is P-selectin now on the surface of the endothelial cells. Now, the other thing that these endothelial cells start to put on their surface is they start to produce something known as platelet activating factor. Now, this is a lipid molecule, so it's not really on the surface. Instead, it's actually within the phospholipid bilayer. So, platelet activating factor, often abbreviated to PAF for short. So, the long name, the full name, is platelet activating factor, and as I say, it's a lipid molecule, so it's within the phospholipid bilayer. Now, normal cells do not have platelet activating factor within their phospholipid bilayer, but when they undergo type 1 activation, they start producing this platelet activating factor, and it's produced very, very rapidly, basically. It's not like proteins where, you have to undergo transcription of the DNA, then translation of the mRNA, then post-translational modifications, and finally package it all up and send it to the membrane. It's not like that. It's a very quick production. Okay, so this is produced on the spot in type 1 activated endothelial cells. Now, let's discuss how we recruit neutrophils now, because we've got well, actually, we need to talk about one more molecule that is on the surface of the endothelial cells. Now, this is constitutively expressed on the surface of the endothelial cells. Okay, so it's on all endothelial cells within your body, and its name is the intercellular adhesion molecule 1. Okay, so as we'll see in a moment when we discuss type 2 activation, you do upregulate this molecule in inflammation, but it is always expressed on the surface of your endothelial cells, always. Okay, so intercellular adhesion molecule 1. Okay, and for short, this is abbreviated to ICAM1. Okay, so let's see the process now by which we extravasate neutrophils. Okay, so the first process is what's known as rolling. So let me explain what happens here. So, actually, maybe we should discuss the structure of a neutrophil first, otherwise I'm just going to draw a picture of a neutrophil. It's going to look a little bizarre. Okay, so before we discuss rolling, let's put a picture of a neutrophil. Okay, so I might put this neutrophil here so that it will be conveniently positioned for my next drawing. Okay, so this round blob is going to represent a neutrophil. So a neutrophil is a type of white blood cell. Okay, so it's a leukocyte, which is just a fancy word for a white blood cell. And neutrophils are a phagocyte, basically, and they're very, very highly present in the blood. So you have a large population of them within the blood. Now, another fancy thing about them is that they have this multi-lobe nucleus that I've drawn here, okay? And they don't have multiple nuclei. I just want to stress this. This is not more than one nuclei. No, they have one nucleus. It's just got this bizarre shape where it has lobes and then little tubes connecting the lobes, okay? And the number of lobes that a neutrophil nucleus has is not set, so it can vary. Now, the archetypal number for a neutrophil nucleus to have is around 3, which is why I've drawn 3, but you can have them with 4, 5, 6 even, and for that reason they're often known as polymorphonucleocytes, okay, which means many shaped nucleus cells, basically, nucleocytes, okay, and the abbreviation for polymorphonucleocytes is PMNs for short. Right, so these cells are a phagocyte. Once they're recruited into the interstitial fluid, they'll start phagocytosing the pathogens and destroying them, hopefully. Okay, but first we have to get this neutrophil from the bloodstream into the interstitial fluid. Okay, so how do the type 1 activated endothelial cells do this? Well, it's going to involve all of these molecules here. Now, the first one is p-selectin, and what's going to happen first is a process of rolling. So basically, what's going to happen is this neutrophil that's within the blood is going to start rolling over the endothelial cells, and as it rolls, what will be happening is it will continuously be forming interactions between p-selectin and a ligand for p-selectin that it has on its surface. So neutrophils always have on their surface a ligand for p-selectin, and this is rather sensibly known as the p-selectin glycoprotein ligand 1. Okay, and because p-selectin glycoprotein ligand 1 is a bit of a mouthful, people often abbreviate p-selectin glycoprotein ligand 1 to PSGL1. Okay, so neutrophils always have PSGL1 on their surface. It's the endothelial cell that has changed, remember. Endothelial cells don't usually have p-selectin on their surface. Okay, so here's the p-selectin in red, and the p-selectin glycoprotein ligand 1 is in blue. Okay, so upon type 1 activation, this endothelial cell has fused the viable palade bodies with the apical membrane, and it now has p-selectin on its surface, which will bind with the p-selectin glycoprotein ligand 1 on the surface of the neutrophil. Now you might think, well, isn't this just going to hold the neutrophil onto this endothelial cell? And no, it doesn't. It's not quite strong enough for that. Instead, what it causes is the rolling of the neutrophil over the endothelial cell. So what will happen is this neutrophil will temporarily form a bond with the p-selectin via its p-selectin glycoprotein ligand 1, but then it's still feeling enough force forward, it's still got enough momentum forward to break this bond, and then what will happen is it will have another p-selectin glycoprotein ligand 1 here. And as this thing rolls, can you imagine what's going to happen here? This one's going to break, okay? And this is going to come down and it will form a new interaction with this one. So let me draw the kind of next stage along, if you like. Okay, so here's our endothelial cell, drawn again. Okay. Here are its two p-selectin molecules, and of course it will have far more than 2, but for the basis of this picture I've just drawn 2, and now the interaction with the neutrophil and the endothelial cell is between this p-selectin glycoprotein ligand and this p-selectin here. Okay, so let me colour them in. So here's p-selectin, the two p-selectin molecules in red. Here are the two p-selectin glycoprotein ligand molecules in blue. Okay, and then we have the multi-lobed nucleus of the neutrophil here. Okay, so you can see that what's happened is this one has broken and it's rolled over and formed a new interaction between this second p-selectin glycoprotein ligand 1 and the next p-selectin. And then of course there'll be another endothelial cell along here, okay? And this endothelial cell will have another p-selectin on its surface, so let's put another p-selectin here. Let's just colour in the nucleus of that endothelial cell. And of course the neutrophil will have another p-selectin glycoprotein ligand 1 here. And what will happen is that this bond between the p-selectin glycoprotein ligand 1 and the p-selectin will cleave, and the neutrophil will form a new interaction between this p-selectin glycoprotein ligand 1 here and this p-selectin on this next endothelial cell. Okay, so that's the process of rolling. So that's the first part of neutrophil extravasation in response to type 1 activation. So you'll be rolling and continuously forming these interactions between p-selectin glycoprotein ligand 1 and p-selectin. Okay, now this has the effect of gradually slowing the neutrophil down. And then the next step, what's going to happen next is that you're going to get a weak adhesion between the neutrophil and the endothelial cell. So let me show you what's going to happen now. So eventually the neutrophil will come to a halt. Okay, so here is our endothelial cell. Here is its nucleus here. And now the neutrophil has come to a halt. And the reason it's come to a halt is that you've formed another interaction between this endothelial cell and this neutrophil. So let's put our neutrophil here. Again, it's getting a little squashed simply for lack of space. And you've still got this interaction between p-selectin glycoprotein ligand 1, which I might just outline in blue rather than colouring in fully blue now. And also p-selectin on the surface of the endothelial cell. So you've still got this interaction, but now you've got an additional interaction. An interaction between ICAM1 here and its ligand on the surface of the neutrophil, which is known as LFA1. Okay, so let me colour this one in orange here. So let me just put in the nucleus of the neutrophil again, this multi-lobe nucleus here to make the picture complete. And now I'll label up these new, this new member here, this LFA1. So LFA1 is another type of cell adhesion molecule. And I should have said this, ICAM1 is a cell adhesion molecule. So remember, in turquoise, this ICAM1, which is always expressed to some level on endothelial cells, this stands for Intercellular Adhesion Molecule 1. Now, it's a cell adhesion molecule within the family of immunoglobulin superfamily cell adhesion molecules. So there's a huge group of proteins in biology known as cell adhesion molecules, and in the family of cell adhesion molecules there are 4 distinct families basically. So there are the immunoglobulin superfamily cell adhesion molecules, which ICAM1 is an example of. Then there is the integrin family of cell adhesion molecules, which LFA1 is an example of. Okay, then there's the selectin family, which p-selectin is an example of. And then finally there's also the cadherin family, which we haven't seen any examples of here. Okay, so LFA1 stands for the lymphocyte, that's the L in its name, function-associated antigen 1. So F stands for function associated, so it's these 2 words that are linked together by that dash. And then the final A stands for antigen 1. Now it's an integrin cell adhesion molecule, which means that it consists of both an alpha subunit and a beta subunit. So if we zoom in on the structure of LFA1, then it will actually have 2 proteins making it up. Well, more than 2, because each of these subunits consist of slightly more than 1 protein. Well, some of them do anyway. Okay, so the integrin family cell adhesion molecules, all of them have these 2 subunits, an alpha subunit and a beta subunit. Now, there are 18 different alpha subunits which you can use. So let's say this is the alpha subunit here, and this is the beta subunit. So there are 18 different alpha subunits that exist in the human genome, and there are 8 different beta subunits. Now LFA1 is specifically made up of the alpha L alpha subunit. So this is one of the 18, and then also the beta 2 subunit. Okay, so it's known as the alpha L beta 2 integrin. Okay, right. So you get this weak interaction between ICAM1 and LFA1, and the reason is that this integrin, LFA1, is not activated yet. Okay, so before you activate LFA1, the bond between the ICAM1 and the LFA1 is very weak. So this next stage is what's known as weak adhesion. Okay, now what's going to save the day? Well, basically you're going to get activation of that integrin basically on the surface of the neutrophil. So what's going to happen is platelet activating factor is going to lead to the activation of that integrin. So here is platelet activating factor, which we'll draw in blue here on the surface of the endothelium. Okay, so remember the abbreviation for platelet activating factor is PAF, P-A-F. Okay, and it's going to bind to the platelet activating factor receptor on the surface of the neutrophil. So this is the platelet activating factor, and then you'll just put R afterwards to denote receptor. Okay, and I'll colour this in yellow. So this is the platelet activating factor receptor in yellow here, and basically when platelet activating factor binds to the platelet activating factor receptor on the surface of the neutrophil, that is going to lead to downstream pathways within the neutrophil which activate the LFA1 integrin, so the lymphocyte function associated antigen 1. Okay, so the next step of what you're going to get is integrin activation, which leads to this adhesion between the LFA1 and the ICAM1 becoming much stronger. So you go from having a very weak adhesion to having a tight bond or a tight adhesion. Okay, and it's because of the activation of the integrin. So what takes you from the weak adhesion to the tight adhesion is activation of the integrin. So this is integrin activation, which is undertaken by the platelet activating factor binding to the platelet activating factor receptor on the surface of the neutrophil. Okay, then finally the next process in this leukocyte extravasation is to actually move the neutrophil into the interstitial space. Okay, so what's going to happen is a process known as endothelial transmigration, also called diapedesis. Okay, so let me show this. So what happens is the neutrophil is going to deform itself to try and fit through the gap between two adjacent endothelial cells. So here we have two endothelial cells, and we know that they've undergone type 1 activation, and therefore they have opened up this gap between the two endothelial cells. And now what's going to happen is that this neutrophil is going to try and slither like a snake through this gap, and this deformation in its shape that it undertakes to get through this gap is what's known as diapedesis. So this process of slithering through, deforming yourself so that you can slither through that gap is known as diapedesis, or also as endothelial transmigration. Okay, so I'll write that down as well. So it's also sometimes referred to as endothelial transmigration. Okay, now there is an important interaction here that occurs within the diapedesis. Let me just go over this. So endothelial transmigration. Okay, so basically there is another cell adhesion molecule which is within the family of immunoglobulin superfamily cell adhesion molecules, which plays an important role in this diapedesis of neutrophils out of the blood vessel. Okay, so basically when you have two endothelial cells that are initially stuck together, I told you about the tight junctions and I told you about the adherens junctions, but there is also another type of junction that's even below the adherens junctions. Let's let this be the tight junction up here. Let's let this be the adherens junction here, and below that there's another type of little junction between the two cells that's made up of these two cell adhesion molecules of the immunoglobulin superfamily cell adhesion molecule type that are bound to each other. Okay, and this cell adhesion molecule that each cell has on its surface is what's known as PCAM1. Okay, so this stands for platelet, that's the P, and then the E is for endothelial. Okay, so you have a dash and then endothelial, and then cell adhesion molecule 1. Okay, so cell adhesion molecule 1. So the platelet endothelial cell adhesion molecule 1 is forming these junctions between endothelial cells. Okay, so it's expressed on the surface of the endothelial cell that's in contact with this next endothelial cell along. Now, PCAM1 is also known by the name CD31, and you do often hear it called that. Okay, so it's worth being aware of. So each endothelial cell has a molecule of PCAM1 on its surface, and the two molecules bind together. Now, when you undergo endothelial contraction, these PCAM1 molecules will be broken apart, and now when the neutrophil moves through, it also has PCAM1 molecules on its surface. So what you'll get basically is an interaction between the neutrophil membrane here, so let's say this is the neutrophil membrane and this is the endothelial cell membrane, so you'll have PCAM1 on both cells binding to one another. And this is this is thought to be a very important interaction for helping the neutrophil through that gap between the endothelial cells. So PCAM1 molecules on the surface of the neutrophil interact with PCAM1 molecules on the surface of the endothelial cell as the neutrophil diapedeses through that gap. Then you have the neutrophil in the interstitial space, and there it will begin the process of phagocytosing the pathogens. Okay, so this is the process by which you get neutrophil extravasation in response to type 1 activation of endothelial cells. However, once type 2 activation has been initiated by interleukin-1 and tumor necrosis factor alpha, you also get another type of neutrophil extravasation, and it's the same basic principles but different molecules, and you also begin to get monocyte extravasation. So we'll continue this discussion where we'll talk about type 2 activation and then how it leads to slightly different neutrophil extravasation and then how it leads to monocyte extravasation. Okay, in the next video.